Hello there, and welcome to the Sleepy Science Society. Tonight, we are drifting into the quiet, mysterious world of mushrooms. These remarkable organisms have been with us since before we walked the earth, yet most of us know almost nothing about them. If this gentle journey through fungal wonders brings you peace, I invite you to like, subscribe, or share your thoughts below. It helps others find their way to this restful corner of science, one sleepy soul at a time. But for now, all you need to do is settle in. Let your shoulders drop. Allow your breathing to slow, and let your mind wander into the fascinating hidden realm that exists right beneath our feet. When you see a mushroom pushing up through soil after an autumn rain, you are witnessing only the smallest visible fraction of something much larger. The mushroom itself, the part we can see and touch, is merely the fruiting body of an organism that exists almost entirely underground. Think of it like an apple on a tree. The apple is just the reproductive structure, the way the tree spreads its seeds. In the same way, a mushroom is simply the spore-producing structure of a vast network that sprawls through the earth beneath your feet. That network is called mycelium, and it is composed of thread-like structures called hyphae that weave through soil, wood, and organic matter in patterns so intricate they resemble neural networks in a brain. These threads are incredibly thin, often just a few micrometers in diameter, yet they can extend for miles. In a single gram of forest soil, there can be several meters of these fungal threads, quietly connecting, decomposing, and facilitating life in ways we are only beginning to understand. The organism you see as a small cluster of mushrooms might actually span acres underground. In eastern Oregon, scientists discovered a single individual honey fungus, Armillaria ostoyi, that covers roughly 2,384 acres of forest floor. This organism has been growing for an estimated 2,400 years, making it one of the largest and oldest living organisms on Earth. It survives by feeding on both living and dead wood, its mycelium spreading slowly but relentlessly through the soil, connecting trees and logs across an area larger than many small towns. When you walk through that forest, every step you take might be above this ancient, sprawling creature. Yet you would never know it was there unless it decided to send up mushrooms. Those temporary fruiting bodies that emerge to release spores into the wind. This hidden nature is true of most fungi. What we call a mushroom is just the tip of something far more complex. The mycelium can persist for years, decades, even millennia, while mushrooms appear only briefly when conditions align perfectly. They require the right temperature, the right moisture levels, and the right chemical signals from their environment. When those conditions arrive, the mycelium gathers its resources and pushes upward with astonishing speed. Some mushrooms can grow several inches in just a few hours, their cells rapidly filling with water to create the hydraulic pressure needed to break through soil or even lift paving stones. This rapid expansion is one of the most remarkable growth rates in nature, transforming invisible threads into visible structures almost overnight. Then, just as quickly, the mushrooms release their spores and begin to decay, returning nutrients to the soil and leaving the mycelium to wait for the next opportunity to reproduce. Understanding what mushrooms truly are requires recognizing that fungi occupy their own kingdom of life, distinct from plants, animals, and bacteria. For centuries, fungi were classified as plants because they seemed rooted in place and grew from the ground. But genetic analysis has revealed something surprising. Fungi are actually more closely related to animals than they are to plants. Like us, they are heterotrophs, meaning they cannot produce their own food through photosynthesis. Instead, they must consume organic matter to survive. Unlike plants, which absorb sunlight and carbon dioxide to create sugars, fungi secrete digestive enzymes into their surroundings, breaking down complex organic molecules externally before absorbing the resulting nutrients. They digest their food outside their bodies, essentially eating the environment around them. This fundamental difference in how they obtain energy places them in an entirely separate category of life. Fungi also have cell walls made of chitin, the same tough, flexible material that forms the exoskeletons of insects and crustaceans. This is another trait they share with animals rather than plants, whose cell walls are made of cellulose. This discovery reshaped our entire understanding of the tree of life. Mycology, the study of fungi, was once considered a branch of botany, but since the late 20th century, it has been recognized as its own distinct field, with fungi deserving their own kingdom. There are currently around 144,000 known species of fungi, but scientists estimate there may be as many as 3.8 million species still undiscovered. This means we have identified less than 5% of the fungal diversity on Earth. Every year, researchers describe thousands of new species, and each discovery reveals more about the astonishing complexity of this often overlooked kingdom. The history of fungi stretches back further than most people realize. The best current estimates suggest that the fungal kingdom is over 1 billion years old. Fossil evidence shows that fungi were among the earliest complex life forms to colonize land. Some researchers have even found structures in rock formations dating back 2.4 billion years that resemble fungal mycelium, though whether these ancient fossils represent true fungi or simply similar organisms remains a matter of scientific debate. What is certain is that fungi played a crucial role in shaping life on Earth. Before plants could colonize land, the terrestrial environment was harsh, barren rock with little organic matter and no soil as we know it today. Fungi were among the first organisms to break down rock and create the conditions necessary for other life to follow. Around 450 million years ago, plants began their transition from water to land. This was one of the most significant events in the history of life, but plants could not have done it alone. Fossil evidence shows that the earliest land plants formed partnerships with fungi almost immediately. These ancient mycorrhizal relationships, symbiotic associations between fungal hyphae and plant roots, allowed plants to access water and nutrients from the soil far more effectively than their roots could manage on their own. In return, the plants provided the fungi with sugars produced through photosynthesis. This partnership was so successful that it enabled plants to spread across continents, eventually transforming the planet's atmosphere and climate. Even today, roughly 90% of all land plants maintain mycorrhizal relationships with fungi. These partnerships are not optional extras, but fundamental requirements for most plant life. Without fungi, the green world we take for granted simply would not exist. Fungi contributed to the formation of soil itself. As they broke down rock and organic matter, they created the rich, complex medium in which plants could root and thrive. They also played a role in one of Earth's great climatic events. During the Carboniferous period, around 300 million years ago, vast forests covered much of the planet. Trees grew enormous, but the fungi capable of decomposing wood had not yet fully evolved. As a result, dead trees did not rot away quickly. Instead, they piled up layer upon layer, eventually forming the coal deposits we extract today. It was only later, when white rot fungi evolved the enzymes capable of breaking down lignin, the tough structural component of wood, that this accumulation stopped. These fungi, with their ability to decompose even the most resistant organic materials, became essential regulators of the carbon cycle. They prevented the planet from being buried under its own growth. This history reveals fungi as quiet architects of the world. They do not photosynthesize or hunt. They do not move through space in ways we can easily see. Yet their influence is everywhere. They shape ecosystems, regulate nutrient cycles, and form partnerships that underpin the survival of nearly all terrestrial life. When you look at a forest, you are seeing the visible result of countless invisible fungal interactions. The trees stand tall because fungi deliver water and minerals to their roots. The soil is rich because fungi break down fallen leaves and logs. The air itself has been shaped by the way fungi manage carbon, releasing and storing it through decomposition. In this quiet, patient way, fungi have been shaping Earth for over 1 billion years. Beneath every forest floor, beneath every meadow and grassland, lies an intricate network of fungal threads that connects plants to one another in ways that have only recently come to light. This network is called a mycorrhizal network, and it has been poetically dubbed the Wood Wide Web, a term that captures both its complexity and its communicative function. The discovery of these networks has fundamentally changed how we understand forests. For a long time, ecologists viewed trees as largely independent organisms, competing with one another for sunlight, water, and nutrients. But research over the past few decades has revealed that trees are far more interconnected than anyone imagined. They share resources, send warning signals, and even support one another through difficult times, all mediated by the fungal networks that link their roots underground. The scientist most associated with this discovery is Suzanne Simard, a professor of forest ecology at the University of British Columbia. Simard grew up in the forests of Canada, where her family worked as loggers. She watched as clear-cutting and replanting practices created plantations of uniform trees, and she noticed that these plantations did not thrive the way natural forests did. Trees planted in rows, separated from the diverse plant communities they would naturally grow alongside, often grew slowly and struggled with disease. This observation led Simard to investigate what was happening below ground. In the late 1990s, she conducted experiments using radioactive carbon isotopes to trace the movement of sugars between trees. She found that carbon was flowing from one tree to another through the soil, and the pathway was fungal hyphae. Trees were not just taking from the fungi. They were using fungi as intermediaries to share resources with one another, Simard's research revealed that older, larger trees, which she calls mother trees, play a particularly important role in these networks. Mother trees are the biggest, most established individuals in a forest, and they often have the most extensive fungal connections. Their roots reach deep into the soil, accessing water and nutrients that younger trees cannot yet reach. Through the mycorrhizal network, mother trees can transfer some of these resources to seedlings growing in the shade of the canopy, giving young trees a better chance of survival. This is not altruism in the human sense, but it functions as a form of resource sharing that benefits the forest community as a whole. The fungi benefit, too, because by keeping more trees alive and healthy, they ensure a steady supply of the sugars that fuel their own growth. It is a three-way partnership, a mutualistic relationship where trees, seedlings, and fungi all gain something essential. The mycorrhizal network does more than move nutrients. It also transmits chemical signals. When a tree is attacked by insects or infected by a pathogen, it produces defensive chemicals within its own tissues. But it also releases chemical signals into the soil, and these signals can travel through the fungal network to neighboring trees. When other trees receive these signals, they begin ramping up their own defenses, even before the threat reaches them. They produce more tannins, compounds that make their leaves less palatable to insects. They alter the chemistry of their sap. They thicken the protective layers on their leaves. This advance warning system allows trees to prepare for attack, reducing the damage they suffer and increasing their chances of survival. Studies have shown that trees connected to mycorrhizal networks respond more quickly and effectively to threats than trees that are isolated from the network. This communication is not fast by human standards. It does not happen in milliseconds like nerve impulses. Instead, chemical signals move through the soil and through fungal hyphae at a pace that might take hours or days. But in the timescale of a tree's life, this is still remarkably quick communication. It allows forests to function as coordinated communities rather than collections of isolated individuals. The mycelium acts like a slow, distributed nervous system, connecting members of the forest and allowing them to respond collectively to changes in their environment. This has profound implications for how we think about intelligence and behavior in organisms that lack brains. The forest, in a very real sense, behaves as a single interconnected entity, with the mycelium serving as the communication infrastructure that makes that coordination possible. The fungi themselves are not passive conduits. They actively regulate the flow of nutrients through the network, allocating resources in ways that benefit their own survival. Some research suggests that fungi can favor certain plants over others, providing more nutrients to individuals that supply them with more sugar, while restricting resources to plants that are less generous. This creates a kind of market system underground, where the currency is carbon and minerals, and the fungi act as traders, brokers, and sometimes gatekeepers. The relationship between plants and fungi is not always harmonious. It exists on a spectrum from mutualism, where both partners benefit, to parasitism, where one partner exploits the other. The exact nature of the relationship can shift over time, depending on environmental conditions, the health of the partners, and the availability of resources. In times of plenty, the relationship is more mutualistic. Times of scarcity, it can become more exploitative. This dynamic balance is part of what makes these networks so resilient and adaptable. Research into mycorrhizal networks has also revealed that plants can recognize their relatives. Trees growing near genetic kin tend to share more resources through fungal networks than trees growing near unrelated individuals. This kin recognition may be mediated by chemical signals released into the soil or by the specific microbial communities associated with each plant's roots. The mechanisms are not yet fully understood, but the implications are striking. Forests are not simply random assemblages of trees. They are communities with social structures where relatedness and history influence behavior. Mother trees invest more heavily in their offspring, increasing the likelihood that their genes will persist into future generations. This challenges the traditional view of natural selection as purely competitive. Cooperation, facilitated by fungi, is also a powerful evolutionary strategy. The wood wide web is not limited to a single forest or a single species of fungus. Mycorrhizal networks can span hundreds of trees and include multiple species of both plants and fungi. In a study of Douglas fir forests in Canada, researchers found that a single fungal network could connect up to 47 different trees. Some networks extend for miles, linking entire hillsides into a single, interconnected system. The scale of these networks is staggering. The total length of fungal hyphae in a forest can exceed the length of all the fine root hairs of all the trees combined. The mycelium is the dominant biological structure in the soil, and it is constantly growing, branching, dying back, and regrowing in response to the needs and signals of the plants it serves. This makes the soil not a static medium, but a living, dynamic network, constantly reshaping itself in response to the activities of the organisms that inhabit it. Understanding these networks has practical implications for forestry, agriculture, and conservation. Clear-cutting forests does not just remove trees. It destroys the mycorrhizal networks that support them. When forests are replanted after logging, the absence of these networks means that young trees must start from scratch, forming new fungal partnerships and slowly rebuilding the underground infrastructure. This is one reason why planted forests often take much longer to mature and are more vulnerable to disease and environmental stress than natural forests where the networks remain intact. Conservation efforts that protect old-growth forests are not just preserving ancient trees, they are preserving the fungal networks that make those forests resilient and productive. Similarly, agricultural practices that support mycorrhizal fungi, such as reducing tillage and avoiding excessive use of chemical fertilizers, can improve soil health and increase crop yields over the long term. The discovery of the wood wide web has also inspired new ways of thinking about communication, cooperation, and intelligence in the natural world. If trees can share information and resources through fungal networks, then forests can be understood as superorganisms, entities that function as coherent wholes rather than collections of independent parts. This view challenges the Western scientific tradition of analyzing nature by breaking it down into isolated components. It suggests that to truly understand ecosystems, we need to look at the relationships between organisms, the flows of energy and information that connect them, and the emergent properties that arise from those connections. The mycelium is a perfect example of this. It is not a single thing, but a network, and its properties, its ability to transport nutrients, transmit signals, and regulate flows, emerge from the collective behavior of countless individual hyphae working in concert. Mushrooms come in an astonishing variety of forms, sizes, and colors. The classic image of a mushroom features a cap supported by a stem, with gills radiating underneath the cap like the spokes of an umbrella. This structure is designed to maximize spore dispersal. The gills are lined with specialized cells called basidia, which produce spores through a process that involves tiny, explosive bursts of moisture. When conditions are right, millions of spores are launched from the gills into the air currents beneath the cap, where they are carried away by the wind. A single, large mushroom can release billions of spores over the course of its brief life, saturating the air with microscopic particles that drift on the breeze until they land in a suitable location and begin to grow. Not all mushrooms have gills. Some have pores instead, tiny holes on the underside of the cap that lead to spongy layers of spore-producing tissue. Boletes, a family of mushrooms often found in forests, use this pore structure to release their spores. Others, like puffballs, enclose their spores inside a papery skin that eventually ruptures, releasing a cloud of spores into the air when the mushroom is disturbed by rain, wind, or the footsteps of passing animals. Still others, like the stinkhorns, use an entirely different strategy. Stinkhorns produce a foul-smelling slime that covers their spore-bearing surfaces. This slime smells like rotting meat, and it attracts flies and beetles, which land on the mushroom and inadvertently pick up spores on their bodies. When the insects fly away, they carry the spores with them, depositing them in new locations. This is a form of insect-mediated spore dispersal, analogous to the way flowers use pollinators to spread pollen. Truffles represent another fascinating variation. These underground mushrooms never emerge into the air. Instead, they develop entirely beneath the soil, where they produce intensely aromatic compounds that filter up through the earth and attract animals like pigs, dogs, and squirrels. When an animal digs up a truffle and eats it, the spores pass through the animal's digestive system and are deposited elsewhere in the forest, encased in a packet of nutrient-rich feces that helps the spores germinate. This relationship has been honed over millions of years, with truffles evolving scents that are irresistible to certain animals, and those animals in turn playing a crucial role in the life cycle of the fungus. Humans, of course, have also been drawn to the aroma of truffles, and these mushrooms have become among the most prized and expensive foods in the world. The specific scent of a truffle is a kind of evolutionary portrait, a signal that has been refined through countless generations to ensure that animals will seek out the fungus and spread its spores. The speed at which mushrooms grow is one of their most remarkable features. A mushroom can emerge from the soil, expand to full size, release its spores, and begin to decay within the span of a few days. This rapid growth is made possible by a unique mechanism. The mycelium accumulates nutrients and energy over time, storing them in its underground network. When environmental conditions trigger fruiting, the mycelium rapidly transports these resources to the developing mushroom. The mushroom's cells fill with water, creating immense hydraulic pressure that forces the structure upward through soil, leaf litter, and even hard substrates like wood or asphalt. This pressure can be so great that some mushrooms can push through tarmac or lift paving stones, cracking them in the process. The speed of growth is driven by this hydraulic mechanism, which allows the mushroom to expand without needing to synthesize large amounts of new cellular material. Instead, existing cells simply inflate like balloons, rapidly reaching their final size. Once a mushroom has released its spores, it begins to decay almost immediately. Fungi are masters of decomposition, and they do not spare their own fruiting bodies. Bacteria and other microorganisms colonize the mushroom, breaking down its tissues and returning nutrients to the soil. The mycelium, however, remains alive underground, ready to produce another flush of mushrooms when conditions are favorable again. Some species fruit multiple times in a single season, while others fruit only once every few years. The timing depends on the species, the availability of resources, and environmental triggers like temperature, moisture, and day length. This separation between the persistent mycelium and the ephemeral fruiting body is one of the key features of fungal life. It allows fungi to survive long periods of unfavorable conditions, hidden underground, and to reproduce opportunistically when the moment is right. Mushrooms also exhibit some truly strange growth patterns. Fairy rings are one of the most visually striking examples. These are circular or arc-shaped arrangements of mushrooms that appear in lawns, meadows, and forests. The ring forms because the mycelium grows outward from a central point, spreading in all directions at roughly the same rate. As the mycelium expands, it depletes the nutrients in the soil at the center, and the active growing edge moves outward. Mushrooms fruit at this active edge, forming a ring that grows larger each year. Some fairy rings are centuries old and can be hundreds of feet in diameter. In folklore, these rings were thought to mark the paths of dancing fairies, and stepping inside a fairy ring was said to bring bad luck or transport you to the fairy realm. The real explanation is less magical, but no less fascinating. Fairy rings are the visible traces of fungal growth over time, a slow, patient expansion that can persist for generations. Another remarkable aspect of mushroom reproduction is the complexity of fungal mating systems. Unlike animals, which typically have two sexes, many fungi have thousands of different mating types. Two mycelia can fuse and exchange genetic material only if they are of compatible mating types. This system greatly increases genetic diversity within fungal populations, ensuring that each generation is genetically distinct from the last. When two compatible mycelia meet, they fuse to form a eukaryotic mycelium, a structure in which each cell contains two separate nuclei, one from each parent. This dikaryotic state can persist for years, even decades, with the two nuclei coexisting in the same cells but remaining separate. Only when conditions are right for reproduction do the nuclei finally fuse, undergoing meiosis to produce spores that contain a single nucleus each. These spores then germinate to produce new monokaryotic mycelia, which must find compatible partners to continue the cycle. This complex life cycle allows fungi to maintain genetic diversity while also exploiting environments opportunistically, forming partnerships, and spreading through the soil without needing to reproduce sexually every generation. Fungi are the primary decomposers in most terrestrial ecosystems. Without them, the world would be buried under a steadily accumulating layer of dead plant material. Every leaf that falls, every branch that breaks, every tree that dies, all of this organic matter would simply pile up, locking away nutrients and carbon that other organisms need. Fungi prevent this by secreting enzymes that break down cellulose, lignin, and other complex organic molecules, converting them into simpler compounds that can be absorbed and reused. This process of decomposition is essential for nutrient cycling. Nitrogen, phosphorus, potassium, and other elements that plants need are bound up in organic matter. Fungi release these nutrients back into the soil, making them available for uptake by living plants. This cycle of growth, death, decay, and renewal is fundamental to the functioning of ecosystems. Different fungi specialize in decomposing different materials. Some prefer soft, nitrogen-rich tissues like leaves and herbaceous stems. Others specialize in breaking down wood, which is much tougher and more resistant to decay. Wood-decaying fungi can be divided into two main groups: brown rot fungi, which break down cellulose but leave lignin behind, and white rot fungi, which can break down both cellulose and lignin. White rot fungi are particularly important because lignin is one of the most chemically stable substances produced by living organisms. It is what gives wood its strength and rigidity, and very few organisms can break it down. White rot fungi achieve this by producing powerful enzymes that cleave the complex aromatic rings of lignin molecules. This ability makes them critical players in the carbon cycle because it prevents lignin from accumulating and allows carbon that would otherwise be locked in dead wood to be released back into the atmosphere or incorporated into the soil. Fungal decomposition also plays a role in soil formation. As fungi break down organic matter, they mix it with mineral particles, creating the rich, complex matrix that we call soil. The hyphae themselves contribute to soil structure by binding particles together and creating networks of pores that allow water and air to infiltrate. Healthy soil is full of fungal hyphae, and this fungal biomass is a major component of soil organic matter. When soil is degraded by intensive agriculture, compaction, or erosion, one of the first things to disappear is the fungal community. Without fungi, soil loses its structure, becoming hard and impermeable, and its ability to support plant growth declines. Restoring fungal communities is therefore a key part of efforts to rehabilitate degraded soils and restore ecosystem function. Fungi also contribute to carbon storage. When fungi decompose organic matter, some of the carbon is released as carbon dioxide, but not all of it. Some carbon becomes incorporated into the bodies of the fungi themselves, and some is transformed into stable organic compounds that persist in the soil for years, decades, or even centuries. A substance called glomalin, produced by mycorrhizal fungi, is particularly important in this regard. Glomalin is a sticky protein that coats fungal hyphae and binds soil particles together. It is extremely stable and can account for a significant fraction of the organic carbon in soil. By producing glomalin, mycorrhizal fungi help sequester carbon, removing it from the atmosphere and locking it away in the soil. This makes fungi important players in the global carbon cycle and highlights their potential role in mitigating climate change. Protecting and promoting fungal communities, particularly mycorrhizal fungi, could be an important strategy for enhancing carbon storage in soils. The relationship between fungi and plants goes beyond simple nutrient exchange. Mycorrhizal fungi can also protect plants from pathogens. When fungal hyphae colonize a plant's roots, they form a physical barrier that makes it harder for disease-causing organisms to penetrate. They also stimulate the plant's immune system, priming it to respond more quickly and effectively to infection. In some cases, mycorrhizal fungi produce antibiotics that inhibit the growth of pathogenic bacteria and fungi in the soil. This protective role is one of the reasons why plants with healthy mycorrhizal associations are more resistant to disease and stress than plants without them. It also explains why agricultural systems that support diverse fungal communities tend to be more resilient and productive than those that rely heavily on chemical inputs like fertilizers and pesticides, which can disrupt fungal populations. Fungi also play a role in helping plants cope with environmental stress. During drought, mycorrhizal fungi can extend a plant's reach into the soil, accessing water that the plant's roots alone could not reach. They can also help plants tolerate salty soils by sequestering sodium ions and preventing them from reaching toxic levels in plant tissues. In polluted soils, some fungi can absorb heavy metals and other contaminants, reducing the amount that plants take up. This ability to buffer plants against environmental stress makes mycorrhizal fungi essential partners in a changing world, where plants face increasing challenges from climate change, habitat degradation, and pollution. The diversity of fungi is staggering. Scientists have described around 144,000 species, but estimates suggest there may be between 2 and 4 million species in total. This means that fungi are one of the most diverse groups of organisms on Earth, yet they remain poorly studied compared to plants and animals. New species are being discovered all the time, often in unexpected places. Some fungi live in extreme environments, like the hot springs of Yellowstone, where they survive temperatures that would kill most other organisms. Others thrive in frozen soils in Antarctica, using anti-freeze proteins to prevent ice crystals from forming inside their cells. Some fungi grow inside rocks in desert environments, extracting nutrients from the minerals themselves and forming cryptic communities that are invisible unless you know where to look. One of the most remarkable adaptations in fungi is bioluminescence. Over 110 species of fungi can glow in the dark, producing a greenish light through a chemical reaction similar to that used by fireflies. In some species, only the underground mycelium glows. In others, the mushrooms themselves emit light. The exact reason for this bioluminescence is still debated, but one leading hypothesis is that it attracts insects. The light draws beetles, flies, and other nocturnal insects to the mushroom, and these insects then help disperse the fungal spores. In dense forests, where wind currents are limited, insect-mediated dispersal may be more effective than relying on air currents alone. The glowing mushrooms are particularly common in tropical and subtropical forests, where the humid, still air makes wind dispersal less reliable. Another striking group of fungi are the parasitic species that manipulate the behavior of their hosts. The most famous example is Ophiocordyceps unilateralis, a fungus that infects carpenter ants in tropical rainforests. When an ant is infected, the fungus grows inside its body, gradually taking control of its nervous system. The fungus compels the ant to leave its colony and climb to a specific height on a plant stem, usually around 25 centimeters above the ground. The ant then clamps its jaws onto a leaf or twig in what is called a death grip. Once the ant is in position, the fungus kills it and begins to grow a fruiting body that erupts from the back of the ant's head. This fruiting body releases spores onto the forest floor below, where they can infect new ants. The entire process is timed and coordinated with remarkable precision, ensuring that the fungus disperses its spores in the optimal location and at the optimal height to reach new hosts. This zombie ant phenomenon is not unique to one species. There are many different species of Ophiocordyceps that infect different species of ants, each with slightly different behaviors and preferences. Parasitic fungi are not limited to insects. Some fungi infect plants, causing diseases that can devastate crops and forests. Rusts, smuts, and mildews are all fungal plant pathogens, and they are among the most economically important organisms in agriculture. Wheat rust, for example, has caused famines throughout history by destroying wheat crops. Dutch elm disease, caused by a fungus spread by bark beetles, has killed millions of elm trees in North America and Europe. Chestnut blight, another fungal disease, nearly wiped out the American chestnut, once one of the most common trees in eastern North America. These diseases highlight the destructive potential of fungi, but they also underscore the importance of understanding fungal biology and ecology. By studying how pathogenic fungi spread and infect their hosts, scientists can develop strategies to control them and protect vulnerable plants and ecosystems. Not all parasitic relationships are straightforward. Some fungi exist in a gray area between mutualism and parasitism, shifting between the two depending on environmental conditions. For example, some mycorrhizal fungi can become parasitic if the plant they are associated with is stressed or weakened. Instead of providing nutrients, the fungus may begin to drain resources from the plant, hastening its decline. Conversely, some fungi that are typically considered parasitic can occasionally provide benefits to their hosts. This flexibility is part of what makes fungal relationships so complex and difficult to study. The outcome of a fungal interaction depends on a multitude of factors, including the health of the host, the availability of resources, the presence of other organisms, and the history of the relationship. Understanding these dynamics requires careful observation and experimentation, and it is one of the major challenges in mycology today. Humans have been interacting with mushrooms for as long as we have been human. Archaeological evidence shows that people were collecting mushrooms for food and medicine long before the advent of agriculture. One of the most famous examples comes from Ötzi the Iceman, a 5,300-year-old mummy found in the Alps. Among the items Ötzi carried were two species of fungus. One, a birch polypore, which has medicinal properties and may have been used to treat intestinal parasites or as an antiseptic, and the other, a tinder fungus, used for starting fires. These finds suggest that ancient people had detailed knowledge of the different fungi in their environment and how to use them for various purposes. Mushrooms have been cultivated for food for thousands of years. The most commonly cultivated species today is Agaricus bisporus, the white button mushroom that appears in supermarkets around the world. Interestingly, button mushrooms, crimini mushrooms, and portobello mushrooms are all the same species, just harvested at different stages of maturity. Button mushrooms are harvested when young and white. Crimini mushrooms are allowed to mature slightly longer, developing a brown cap. Portobellos are fully mature, with large, flat caps and exposed gills. This marketing of a single species at different life stages has been highly successful, but it represents only a tiny fraction of the diversity of edible mushrooms. In recent years, more species have become available, including shiitake, oyster mushrooms, lion's mane, and maitake, each with its own flavor, texture, and nutritional profile. These mushrooms are not only delicious, but also nutritious, providing protein, vitamins, minerals, and bioactive compounds with potential health benefits. The global mushroom market is worth tens of billions of dollars, and it continues to grow as more people recognize the culinary and nutritional value of fungi. Mushroom cultivation is relatively sustainable compared to many forms of agriculture. Mushrooms can be grown on agricultural waste products like straw, sawdust, and coffee grounds, converting these materials into high-value food. They require less water and space than conventional crops, and they can be grown indoors, making them suitable for urban agriculture. Some companies are even using mushroom cultivation as a form of bioremediation, growing fungi on contaminated substrates to remove pollutants, and then safely disposing of the mushrooms or composting them. Beyond food, fungi have played a critical role in medicine. The discovery of penicillin, the first widely used antibiotic, is one of the most important medical breakthroughs in history. Penicillin is produced by fungi in the genus Mycelium, and its discovery by Alexander Fleming in 1928 revolutionized the treatment of bacterial infections. Before antibiotics, even minor injuries could lead to life-threatening infections. Penicillin and the antibiotics that followed have saved countless lives. But penicillin is just one example. Many other medically important compounds have been derived from fungi. Cyclosporine, a drug that suppresses the immune system and makes organ transplants possible, comes from a fungus. Statins, widely used to lower cholesterol, were originally isolated from fungal cultures. Numerous other compounds with antiviral, anti-tumor, and immunomodulatory properties have been discovered in fungi, and researchers continue to explore fungal biodiversity in search of new medicines. Some fungi have been used in traditional medicine for centuries. In Asia, mushrooms like reishi, cordyceps, and turkey tail have long been valued for their supposed health benefits. Modern research has begun to investigate these traditional uses, and while the evidence is still emerging, some studies suggest that certain mushrooms may have immune-boosting, anti-inflammatory, or antioxidant properties. The polysaccharides found in mushroom cell walls, particularly beta-glucans, are of particular interest because they appear to modulate the immune system in complex ways. While more research is needed to fully understand these effects, the potential of medicinal mushrooms is an active area of scientific inquiry. Not all mushrooms are benign. Some are highly toxic, and mistaking a poisonous mushroom for an edible one can be fatal. The death cap mushroom, Amanita phalloides, is responsible for the majority of fatal mushroom poisonings worldwide. It looks similar to some edible species, and it can grow in areas where people might not expect to find dangerous mushrooms, such as suburban parks and gardens. The toxins in death cap mushrooms attack the liver and kidneys, and by the time symptoms appear, usually several hours after ingestion, the damage is often irreversible. This highlights the importance of careful identification when foraging for wild mushrooms. Even experienced foragers can make mistakes, and the consequences can be severe. For anyone interested in collecting wild mushrooms, learning from an expert, using multiple sources for identification, and being conservative about what to eat are essential precautions. Fungi can also be psychoactive. Certain species contain compounds like psilocybin, which produces hallucinations and altered states of consciousness. These mushrooms have been used in religious and spiritual ceremonies by indigenous cultures for thousands of years, and they are currently being studied for their potential therapeutic applications. Research suggests that psilocybin, when used in controlled settings with psychological support, may help treat conditions like depression, anxiety, and post-traumatic stress disorder. The way psilocybin works in the brain, by altering serotonin signaling and promoting neuroplasticity, has opened up new avenues for understanding consciousness and mental health. This is a fascinating example of how compounds produced by fungi can have profound effects on human biology and psychology. The study of fungi, mycology, has a rich history. The ancient Greeks and Romans wrote about mushrooms, and the philosopher Theophrastus attempted one of the first classifications of plants and fungi. But it was not until the 18th and 19th centuries that mycology began to develop as a rigorous scientific discipline. Christian Hendrik Persson and Elias Magnus Fries are often considered the founders of modern mycology. Fries developed a classification system based on spore color and the structure of the fruiting body, and many of his classifications are still used today. Miles Joseph Berkeley, a British mycologist, contributed enormously to the field by describing thousands of new species and establishing mycology as a distinct area of study. In the 20th century, advances in microscopy, biochemistry, and genetics transformed mycology. Researchers could examine fungal cells in detail, study the enzymes they produced, and analyze their DNA. DNA sequencing, in particular, has revolutionized our understanding of fungal relationships. Many fungi that look very similar morphologically turn out to be only distantly related, while others that look completely different are close evolutionary cousins. Molecular phylogenetics has revealed that the kingdom fungi is divided into several major groups, with the two largest being the Ascomycota, which includes yeasts and many cup fungi, and the Basidiomycota, which includes most of the mushrooms we are familiar with, as well as rusts and smuts. Within these groups, there is astonishing diversity, and new lineages are still being discovered. DNA analysis has also revealed surprising connections between fungi and other organisms. As mentioned earlier, fungi are more closely related to animals than to plants. This discovery, made in the late 20th century, was one of the most significant findings in evolutionary biology. It meant that fungi and animals shared a common ancestor more recently than either group shared with plants. This ancestor was likely a single-celled organism that lived over 1 billion years ago, and from it evolved two great kingdoms: the animals, which became mobile and developed complex multicellular bodies, and the fungi, which remained anchored in place but evolved the ability to digest the world around them. This deep evolutionary connection means that fungi and animals share many cellular and molecular features, which has implications for medicine, biotechnology, and our understanding of life's history. One of the most exciting areas of fungal research today is bioremediation, the use of fungi to clean up polluted environments. Fungi have an extraordinary ability to break down complex organic molecules, and some species can even degrade plastics, petroleum products, and toxic chemicals that other organisms cannot touch. For example, a fungus called Aspergillus tubangensis has been found to break down polyurethane plastics, which normally take decades or even centuries to decompose. The fungus secretes enzymes that cleave the chemical bonds in the plastic, turning it into simpler molecules that the fungus can absorb and use as food. If this process can be scaled up, it could provide a way to deal with the massive amounts of plastic waste that accumulate in landfills and oceans. Other fungi have been shown to absorb heavy metals from contaminated soil. Some species of oyster mushrooms, for example, can accumulate cadmium, lead, and mercury in their tissues, effectively pulling these toxic metals out of the soil. After the mushrooms are harvested, they can be safely disposed of or even processed to recover the metals. This approach has been tested in polluted industrial sites and has shown promising results. Fungi have also been used to clean up oil spills. After the Exxon Valdez oil spill in Alaska, researchers applied fungi to contaminated beaches and found that the fungi accelerated the breakdown of oil, reducing the time it took for the beaches to recover. Similar approaches have been used after other environmental disasters, including the Fukushima nuclear accident in Japan, where certain fungi were found to be unusually resistant to radiation and were able to break down radioactive compounds. The ability of fungi to tolerate and even thrive in extreme conditions has led to interest in using them in industrial applications. Fungi are already used in the production of many foods and beverages, including bread, beer, wine, cheese, soy sauce, and tempeh. Yeasts are essential for fermentation, the process that converts sugars into alcohol and carbon dioxide. Different strains of yeast produce different flavors and textures, which is why there is such diversity in beers and wines from around the world. In cheesemaking, fungi like Penicillium roqueforti and Penicillium camemberti give blue cheese and camembert their distinctive flavors and textures. In Japan, the fungus Aspergillus oryzae, known as koji, is used to ferment soybeans into soy sauce and miso, and to convert rice starch into sugars for sake production. These traditional uses of fungi highlight how deeply intertwined human culture and fungal biology have become. More recently, fungi have been explored as a source of sustainable materials. Mycelium can be grown into specific shapes and then dried to create lightweight, strong, and biodegradable materials. Companies are using mycelium to make packaging materials that can replace polystyrene foam. The process involves growing mycelium on agricultural waste in molds, then drying and heat treating the material to kill the fungus and prevent further growth. The resulting material is compostable and has good insulating properties. Mycelium-based materials are also being used to create leather substitutes, building materials, and even furniture. As concerns about sustainability and environmental impact grow, fungi offer a promising alternative to synthetic materials derived from fossil fuels. Fungi are also being explored as a source of biofuels. Some fungi can produce enzymes that break down cellulose from plant material into sugars, which can then be fermented into ethanol. This process, known as cellulosic ethanol production, has the potential to turn agricultural waste like corn stalks and wood chips into fuel without competing with food crops. While the technology is still being developed, fungi could play a key role in creating a more sustainable energy system. Their ability to break down tough plant materials efficiently makes them ideal candidates for bioprocessing applications. The study of fungi also has implications for understanding life beyond Earth. Fungi's ability to survive in extreme environments, their efficiency at extracting nutrients from minimal resources, and their resilience to radiation make them potential candidates for supporting human life in space. Researchers have proposed using fungi to recycle organic waste on long-duration space missions, to produce food, and even to help terraform other planets by breaking down rock and creating soil. While these ideas are still speculative, they illustrate how versatile and adaptable fungi are and how much we still have to learn about their capabilities. Fungi challenge our assumptions about what life can be. They are neither plant nor animal, yet they are essential to both. They are mostly hidden, yet their influence is everywhere. They communicate, cooperate, compete, and adapt in ways that reveal intelligence emerging not from brains but from networks. They have shaped the history of life on Earth, and they continue to shape ecosystems, climates, and human societies today. The more we learn about fungi, the more we realize how much we have yet to discover. There may be millions of species still unknown to science, each with its own unique biology, ecology, and potential applications. Exploring this diversity is one of the great frontiers in biology, and it promises to yield insights not only into fungi themselves, but into the fundamental principles that govern life. As you lie here, drifting towards sleep, consider the quiet world beneath your feet. Imagine the mycelium spreading through the soil, connecting trees, recycling nutrients, storing carbon, and communicating in chemical whispers that we are only beginning to understand. Imagine the mushrooms pushing up through leaf litter, releasing billions of spores into the breeze, each one a potential new beginning. Imagine the ancient partnership between fungi and plants, a relationship that made it possible for green life to colonize the land and for forests to spread across the continents. Imagine the decomposers breaking down the dead, transforming what was into what will be, ensuring that nothing is ever truly lost. Fungi are quiet architects, invisible engineers, silent communicators. They shape the world in ways we rarely notice, yet without them, the world would be unrecognizable. If you enjoyed this gentle journey through the fungal kingdom, I invite you once more to like, subscribe, or share your thoughts below. Your support helps others discover these restful explorations of science, and it keeps this little corner of calm alive. For now, let your mind settle. Let the images of mycelium and mushrooms fade into soft shapes at the edges of awareness. You do not need to hold on to any of it. The night is here to cradle you. Your thoughts can slow. Your breathing can deepen. Everything you have learned tonight will find its place in memory, weaving itself into the quiet patterns of your mind as you sleep. There is nothing more you need to do. Simply rest. Simply breathe. Simply let go. The world of mushrooms will still be there tomorrow, hidden and patient, waiting beneath the soil. Good night.